Genetics is a pretty cool field, but it took some thinking to discover what it was in the first place. For that, we have to thank Gregor Mendel. So let's talk about how we discovered genetics in the first place. We have these lovely plants to thank. Genetics was discovered by Gregor Mendel. He was an Austrian monk, and of course, we do consider him the father of genetics. He did a bunch of experiments on the common garden pea. In where he was a monk, he just had a big greenhouse and he had tons of different plants. He would meticulously grow them and breed them. And one of the cool things about peas is they are self fertilizing. So it is really easy to make sure that you understand exactly who are the parents of each individual pea plant. One of the saddest things, or things that I find the saddest about Mendel, is he published his results in a rather obscure journal at the time. And nobody really paid much attention to his findings until long after he was dead. He was rediscovered in 1900. So even though he was a contemporary of Darwin, Darwin didn't know about his research. One of the things that's the biggest what ifs in the history of evolutionary biology is it was later discovered that the journal that Mendel published in was actually in Darwin's office, but it was uncut. At the time, when you would buy books or journals, they would be bound together and you like literally had to cut them open. So that meant Darwin never read it. So it was so close, but so far. But enough of that. Let's talk about what Mendel actually did. So when you look at pea plants, there are a couple different ways they vary. Flowers are either purple or white. Plants are either tall or short. Their seeds are yellow or green. The shape of the seeds are round or wrinkled. The pods can be green or yellow, and those same pods will be either inflated, very puffy, or constricted, so where you can see each of the individual peas. And the flowers are either at the axial, like right where different branches join, or terminal, they're at the end of branches. So you might notice here on all of these traits we're looking at, there are two options. It's one or the other. So you can think of it as kind of a binary choice. So already it's hard to see blending inheritance happen if there are only two choices. But it is a little bit to look at 7 different options. So let's simplify this and let's only think about plant height for a moment. Under the blending model, we would predict that if we have a tall plant and a short plant and we cross them together, we would get all medium plants. And then if we self-fertilize those medium plants, we will get medium plants now until the end of time. And this is one of shows that that the blending model isn't a great model for how we actually see variation in the world, because this would show everything would just go to the exact middle for all traits and we would never get new variation. So that's one chink in the armor of the blending model prediction. But even more so, when we look at the actual results, we see it's a little bit different. When we cross a tall plant and a short plant, all of their offspring are tall. And then when we self-fertilize that first generation, the next generation, we get this really interesting ratio. We get a ratio of 3 tall plants to 1 short plant. Mendel saw this over and over again with all of those different traits he looked at. He was seeing similar types of ratios. And it did take him a little while to figure out what this meant. And to talk about this, he came up with a couple different important terms. The first thing we want to talk about is phenotype. Phenotype is the physical traits that we can observe. So right now, the phenotype is plant height. It can be either tall or short. Another phenotype could be eye color, and the different options are blue or green or brown, any of those. The next part, this is where Mendel was really thinking a little bit more hypothetically. And he didn't know exactly what was going on, but his predictions have held the test of time. What Mendel thought was now invisible to us, we have a genotype. The genotype being that hereditary material that is inside us and inside these pea plants. Hypothetically, he thought, okay, we have 2 parents, so it's getting material from both of its parents, so there's probably 2 parts to this genotype. And if hypothetically we have two different types, we have a dominant and a recessive. And that's how we can end up masking some of these traits. So Mendel came up with the idea that if we have these genotypes that we get from both parents, but one can mask the other, that's where this dominant versus recessive traits come in, that's where we would find this result, or this ratio of 3 tall to 1 short, as long as tall is the dominant trait. And here you can see a classic Punnett square. Along the top, we have the sperm from the dad, so we have a large A and a small a. And on the side, we have the egg from the mom, where we again have a big A and a small a. So if you have two parents that have two different versions of this genotype, this is the ratio you will expect to see in the offspring. So what Mendel was demonstrating here was not blending inheritance. His results did not adhere to those predictions. What he instead saw was particulate inheritance, that we are seeing inheritance happen via particles. So here, this is inheritance of discrete characters that are independently expressed. But let's talk a little bit more about these genotypes, because there's a couple other important words to know. First, we can be homozygous. Again, homo means same. So that means you have two of the same form at whatever gene we're talking about. Alternatively, you can be heterozygous. So that just means you have two different alleles. But next, let's talk about this gene. What is that word we're talking about? You've probably heard it before. When Mendel first talked about this term, he simply meant a molecular unit of heredity. He was not looking at molecular biology, he was only looking at the phenotype. So he was looking at the gross unit and trying to imagine what he thought was inside. A more common term that you might hear today is a specific sequence in the genome that codes for a protein. This can get a little bit complex, and let's use this hemoglobin molecule as our example here. So in this hemoglobin, you see there's 4 different subunits. They are very similar, but they are slightly different. So the question is here, which is the gene? Is it one big gene with 4 separate parts, or do we have 4 closely related genes? So sometimes that line can get a little fuzzy. Also, now there are several important units within the genome that don't necessarily code for a protein. These are primarily important stretches of RNA. So now most people consider a gene to be a specific sequence in the genome that codes for something useful, either a protein or RNA. It's also important to talk about the differences between genes, alleles, and loci. So genes, that is that molecular unit, or that is the trait in question that we are talking about. So going back to our pea plants, that could be height. You could think about it as eye color. The allele, that is the form of the trait. So if we're talking about plant height, the alleles are tall and short. If we're talking about eye color, the alleles are blue or brown. The locus, that just means the physical location. So if we don't really have a specific gene we're talking about, and we're kind of talking about it in the abstract, you might hear people use the word locus or loci, that is the plural. So can you explain? How did Mendel discover genetics?